Hi, I'm Laura Lansdowne, Managing Editor for Technology Networks, and I'm here to introduce our next talk, Targeting the Tumour Microenvironment and the Development of Effective Therapeutics Against Solid Tumours. I'm pleased to be welcomed by Dr. Prajval Paudel. Dr. Paudel received his PhD in biochemistry from University of Delaware, where his research focused on ubiquitin proteasome signaling and de-ubiquitinating enzymes. His expertise ranges from proteomics to enzymology and protein chemistry. Currently, he provides technical expertise to Acro's product and service pipelines and works to communicate Acro's offerings to industry professionals and academia. A warm welcome to you, Prajval. Following the presentation, we will have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss the talk, so feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during the presentation. If you do experience any technical difficulties during the presentation, please request help using the technical help icon in the menu on the left-hand side of the player. I will now hand over to Prajval to begin his presentation.
Thank you. Good afternoon, everyone. First, I would like to thank the organizers of the symposium for a great event. And today I will be presenting on Acro's effort in supporting research to study tumor microenvironment and develop tools and strategies to fight solid tumors. So, although much progress has been made in treating hematological malignancies in the recent decades, solid tumors present a complex challenge, especially in improving efficacy of therapeutics. The tumor microenvironment is comprised of the tumor, the extracellular matrix, and various non-transformed cells, including fibroblasts, immunoinfiltrates, and blood vessels that are recruited from nearby local or distant tissues. The TME plays a key role in tumor initiation, progression, invasion, metastasis, and resistance by deploying matrices, cytokines, growth factors, as well as vascular networks for nutrients and waste exchange. So therefore, understanding the TME requires a close examination at the tissue and cellular level using technologies to resolve and map structures and interrogate signaling pathways. Cells that make up the TME remold and nourish the tumor via multiple cross-talking signaling pathways. Major signaling pathways in the TME include the receptor tyrosine kinase, signaling pathways, steroid hormonal signaling, cytokines, chemokines, and others, as well as the ancient signaling pathways that upregulate or downregulate various pathways impacting tumor progression and therapeutic outcomes. In the past, the cancer research has focused on investigating cellular structures and functions along with genomic expression in mostly homogenized tumor samples. However, recent advancement in single-cell technologies and multiplex immunohistochemistry, as well as technical breakthrough in spatial proteomics and transcriptomics, has enabled the ability to answer fundamental questions in cancer biology, including the interaction of both malignant and non-malignant cells and its functional consequences in cancer progression and therapeutic modalities. Spatial imaging of the TME and especially proteomics has enabled high-resolution reconstruction of the TME. Iterative process like the multiplex immunohistochemistry involving sequential antibody staining and detection enables characterizations of up to 30 markers in a tissue samples, while DNA barcoding methods like Codex enables a complete 3D reconstructions of a tissue section with the detection of more than 50 proteins at a time. Mass spectrometry methods can perform single-cell analysis of up to 34 parameters or up to 40 proteins in tissues using approaches like multiplexed ion beam imaging technique, while UV photocleavable oligonucleotide conjugated antibodies have been used to spatially resolve protein expression in tumor tissue samples with multiplex nanostring DSP technology. And there's no doubt we will hear more about these exciting technologies during this symposium as well. And Acro is committed to providing high-quality protein involved in the TME signaling pathways to accelerate the pace of research and discovery. And today I will go over our TME product line and how we have helped researchers from around the world in understanding the TME and developing better tools to detect and treat solid tumors. First, I will begin with the receptor tyrosine kinase signaling pathway, which are a class of cell surface transmembrane receptors that form cross-linked dimers upon ligand binding leading to activation of tyrosine kinase domain via cross-phosphorylation, followed by intracellular signal transduction. The RTKs bind to growth factors, hormones, and cytokines to elicit a variety of major cellular processes and also play a key role in the tumor microenvironment. Some of the major receptors family involved in the maintenance of TME include the ErbB family receptors, the fibroblast growth factor receptors, vascular endothelial growth factor receptors, and the hepatocyte growth factor receptors. The ErbB family of RTK receptors comprises of 4 members. These ErbB receptors play a prominent role in cancer as cancer drivers and are heavily targeted in cancer therapeutics. Ligands like EGF and neuregulin bind to ErbB receptors, inducing a formation of a homoheterodimer, leading to activation of intracellular phosphorylation signaling cascade and various tumor outcomes. The EGFR, or the epidermal growth factor receptor, is expressed widely in tissues, but in the TME, mutant EGFR leads to immune escape and immunotherapy unresponsiveness or resistance. EGFR mutations are found in 10 to 15% in Caucasians and up to 50% in East Asian patients in lung cancers. And EGFR signaling axis involves the tumor cells, the endothelial cells, and the bone marrow stromal cells. EGF-like peptides regulate synthesis of angiogenesis growth factors in the tumor cells by acting on endothelial cells and bone marrow stromal cells, which promote maturations of the osteoclasts, leading to bone destruction and metastasis. Bone marrow stromal cells themselves produce EGF ligands acting on both the tumor cells and the endothelial cells. ACRO provides not only human EGFR, but also mouse and macaque. And we also provide that with a variety of tags, including biotin, His, and Lama-FC conjugated EGFR for a variety of applications. In addition to accelerating research and improving results, ACRO offers a coupled EGFR beads for antibody screening, capture, and cell sorting applications. Recently, researchers have developed an antibody drug conjugate targeting tumor cells expressing EGFR and using ACRO's high-quality proteins to perform characterization studies, as shown here. ACRO's high-quality EGFR was also used to develop new radio-labeled antibody for immunohistochemistry. Researchers used the technetium-99 isotope-labeled anti-EGFR K9225 IgG to detect native K9 EGFR in mammary carcinoma tissues. And researchers in Denmark relied on ACRO's EGFR protein to characterize a new bioconjugation technique to label proteins and antibody without genetic modification, enabling a new method of imaging by targeting antigens and cell and tissue surfaces. They exploited the metal binding domains of proteins or antibodies to site-specifically label neighboring free amine, enabling fluorophore conjugation and providing a new method for visualizing cancer cell markers. Other family of the ErbB receptors are the HER2 and HER3 that are most closely associated with breast cancer, but also involved in malignancies of several other cancers. ACRO's high-quality HER2, HER3 proteins have been used by researchers from Daiichi Sankyo and Osaka University to immunize rabbits and develop this single-domain antibodies made up of the rabbit variable heavy chain with a cysteine residue mutation to enable disulfide linkage to stabilize the structure. And several clones of this variable heavy chain showed sub-nanomolar affinity to the HER2 and HER3 targets. Another RTKs are the FGFR family receptors involved in the regulation of cell survival, proliferation, differentiation, and cell motility, particularly during embryogenesis, adult tissue homeostasis, and carcinogenesis. The FGFR mutations are implicated in a variety of cancers. In addition to overexpression, type 1 fusion, there are 2 types of gene fusion mutations. The type 1 fusions is caused by chromosomal translocation in hematological malignancies, while type 2 fusions is caused by chromosomal rearrangement in solid tumors. In the TME, FGF secreted by tumor and stromal cells are involved in immune evasion and angiogenesis. VEGFR regulates angiogenesis and tumor vasculature in the TME as well. VEGFR ligands bind to multiple VEGFR subfamily receptors to induce varying pathways. And then you have the HGFR signaling, which is also a clinically important therapeutic target as a proto-oncogene, and promotes development and progression of liver, lung, colon, breast, ovarian, prostate, and gastric carcinomas. Inhibitors targeting the intracellular kinase domain, as well as the extracellular ligand binding SEMA domains, are in clinical trials currently. ACRO offers VEGFR, EGFR, and its ligands, as well as FGFR receptors with His, biotin, Fc, and strip tag with high purity and high bioactivity for R&D applications. At ACRO, we're also proud to have supported basic and translational research to improve understanding of signaling pathways and associated therapeutics. For example, researchers at Yale School of Medicine determined that mice with syndican-2 proteoglycan deletion exhibited angiogenic defects, and using ACRO's VEGF-A165 ligand confirmed direct interaction with the syndican-2 and determined the mechanism of this effect. In another instance, teams from Germany and Yale School of Medicine also developed a VEGF sensor using a luciferase GFP reporter FRET pair for application in monitoring anti-VEGF treatments. To study the TME implication of receptors and to design therapeutics that target downstream signaling pathway, cell-based reporter assay provide a biological model to obtain relevant data. Some transcription factors upregulating cancers are the STATs and the NFATs. The STATs are part of the JAK-STAT pathway, which provide a direct mechanism for extracellular factors to control gene expressions, leading to cancer cell survival and proliferation. NFATs are involved in many aspects of cancer cell proliferations, metastasis, resistance, and development of the tumor microenvironment. And ACRO is developing these STAT3 and NFAT luciferase reporter assay, which will be available in the near future in our catalog. In the development, our ACRO STAT3 reporter assays in HEC293 cells shows high performance upon stimulation with IL-6. Similarly, EGFR reporter assay under the STAT3 control also showed strong specific response to stimulation by EGF. ACRO has developed HEC293 expressing VEGFR-based luciferase reporter assay under the NFAT control, showing high sensitivity and specificity to the VEGF stimulation. Another signaling pathways are the cytokine signaling pathway that are critical in immunosuppression environment of the tumor microenvironment, and also represent an attractive therapeutic target to turn cold tumors into hot tumors. Cytokines have overlapping activities and crosstalk in exerting their effects. And in the TME, cytokines dictate innate and adaptive immune cell responses and regular growth, apoptosis, activation, and differentiation of tumor cells. Cytokine therapies as single agents or combinations are being explored in cancer therapy, exploiting their immunostimulatory and immunosuppressive effects. ACRO provides high-quality interleukins and interferon ligands and receptors to supercharge this research. Additionally, ACRO is also developing GMP-grade IL-7, IL-15 for particular use in cell therapy applications. Here in the table shows some data on our GMP-grade IL-15. And these products will be available relatively soon. Another pathway in the TME is the TGF-beta pathway, which plays a dual role in carcinogenesis, regulating immune and cancer cell activities while promoting tumor suppression in early phases. The TGF-beta pathway switches to pro-invasive function in advanced cancers. The TME is rich in TGF-beta, which is produced by cancer cell fibroblasts, macrophages, platelets, and Tregs, functioning to suppress the immune response against cancer cells. ACRO carries all three TGF-beta ligands and is peptide LAP. For the TGF-beta ligands, both humans and mouse species are available with more than 85% purity and high bioactivity as judged by cell-based assays and binding experiment as well. Another important signaling pathways in the TMEs are integrins. Integrins are a family of cell adhesion receptors controlling cell adhesion to the extracellular matrix. They are comprised of alpha and beta subunits with 24 distinct heterodimers identified to date. Integrins are associated with the receptor tyrosine kinase signaling and promote tumor cell adhesion, angiogenesis, as well as metastasis and tumor aggressiveness. ACRO carries a comprehensive series of integrin receptors and ligands with high bioactivity and batch-to-batch consistency. And ACRO is proud to have supported researchers at Osaka University to develop a radioiodinated bicyclic RGD peptide binder to integrin alpha V beta 3. The specificity of this peptide to the alpha V beta 3 isoform was determined through ACRO's integrin product lines, and labeling of integrin overexpressed in tumor cell line U-H7 compared to non-integrin expressed in A549 tumor cell line shows high performance of this peptide and potential application in clinical setting. The hedgehog, nut, and Wnt signaling pathways are evolutionary conserved pathways that are also upregulated in cancers and regulate genes involved in cell proliferation, survival, migration, and stem cell progenesis. And ACRO carries the catalog of Wnt signaling pathway ligands, receptors, and enzymes, as well as not signaling pathway receptors and ligands. We provide biologically relevant data, including ELISA, BLI, and SPR binding data for all these product lines. So here are some of the key therapies that are currently in clinical trials targeting various aspects of the signaling pathways and markers in the tumor microenvironment. With the improvement in understanding of the TME at tissue level and at molecular level, effective therapies for various solid tumors are within sight. These include targeting the tumor vasculature, tumor-associated macrophages in combination with immune therapeutics, dendritic cells and immune checkpoint therapeutics, and also targeting the extracellular matrix and cancer-associated fibroblast targets. So I would like to conclude that ACRO Biosystem is a premier recombinant protein supplier, as well as providing antibodies, kits, and pre-coupled beads. We have more than 1,600 plus recombinant proteins in our catalog. We use our proprietary HEC293 cell-based protein production platform for our recombinant product lines, and provide a multiplex analytic platform for protein bioactivity testing. To learn more about our products, you can contact us at order@acrobiosystems.com. If you have any questions, you can email me at proswald.podel@acrobiosystems.com, and I'd be happy to take any questions. Thank you.
Thank you very much, Dr. Podel. A fantastic talk. So I anticipate that we're going to get quite a few questions from the audience, and I can see that we've received a couple already. So we'll move straight on to the Q&A now. So our first question for you is, when will the cell-based reporter assay be available, and will they be available as cell lines or as kits?
Right, great question. So the reporter assays mentioned are only part of a more comprehensive pipeline that we have. Currently, we are aiming to create cell lines available to our customers, and we're aiming to develop assay kits in the near future as well. So we have a lot of characterization to do, and we'll keep you all updated on that.
Thank you. Our next question is, you showed a few examples of ACRO proteins being used in developing novel detection and imaging methods. Can you elaborate on how ACRO contributed directly to these efforts?
Right. So what I have shown you today are data from our academic clients. So these data are freely available, are published. But however, our majority of our clients are in the industry space, in small, mid, and large pharmaceutical companies. And we continually work with them, with our industry partners, to develop, conjugated proteins. We work on new methods, provide samples, insights from our technical team, not only to address clients' needs, but also improve their existing workflows and assays. So this is a robust partnership that we're developing. Unfortunately, I'm not able to disclose these efforts, but suffice to say, we have a great working relationship with a lot of our industry partners.
Thank you very much. I think we've got time just for one more question. So the next question is, in what ways do you support collaboration with academia and industry clients?
A great question. So actually, ACRO's vision is to become much more than a vendor of recombinant proteins and kits. We would like to become a close partner in the R&D space to both academia and industry, so that we can accelerate development of novel diagnostics and therapeutics. And to that end, we're always exploring newer technologies and approaches and partners in addressing technical challenges and development hurdles. We're always open to suggestions and improving, or in addition to our product catalog, including mutations, variety of tags and constructs. And we look to new technologies for licensing to bring innovation from university to the commercial space. And we also provide custom CRO services to our small to mid-sized biotech startups, biopharma startups, including providing custom protein production services, SBR and BLI binding services, and as well as cytokine chemokine analysis using MST and ELISA services as well. So we keep our communication open to any our partners in academia and industry to join us in accelerating these great therapeutics and technologies to fight a variety of diseases.
With that, I'd just like to say a huge thank you to our speaker and our audience. We really appreciate your time today. Any questions that we didn't have time to get to will be sent to the speaker after the event. Prajval's talk will be available to view on demand for the next 30 days. We'll be back soon with the next talk.